We have our next fantastic segment that we're going to roll straight into. The next segment that we're having today is Animal Myths, A Bug's Life. So I'd like to welcome our researchers from Macquarie University's Behavioural Ecology Laboratory who look into all various life forms, birds, fish, but especially invertebrates. I'd like to welcome to the stage, can you please make welcome Marilia Fernandez Erickson and Vanessa Pena Gonçalves and Matilda Levia San Silvestri. Welcome. I'll hand over to you.
Hi. Hello, everybody. So, greetings from Behavioral Ecology Lab. We came as a group because we are very social animals. You get to hear you get to hear 4 different talks today, and we try to bust some myths, but really we're just here to tell about our research or prepared. So my specialty is visual ecology, and I study animal vision. I'm specifically interested in dynamicity in visual signals. So if they move, if they flash, what does that do to how we see things? So yeah, straight into it. So we would like to acknowledge the traditional custodians of the land and the people on whose lands we've been collecting data, experimenting data, doing observations, and we would like to pay our respects to the elders, present and future. So I'm going to be talking about my PhD mostly, and specifically of these 3 chapters, not in that order and in a very light manner, but anyway. Just to get you up to speed, visual ecologists have been studying vision of animals for decades now, and we are pretty good at it. We know a lot of things by now. So we know which kind of colors they can see and which kind of visual acuities they might have, which leads to the only myth I'm going to bust here. So we kind of tend to think that animals have a pretty good vision, but that's not true. We should embrace this graph here and take a look at the y-axis. It's actually logarithmic, and humans are right at the top of the list with the eagle, and the rest of the animal kingdom is basically legally blind. So, but this is just human standards, so there's of course a lot more to vision than this. So my research considers how we could think of the dynamicity in vision. So vision consists of 3 dimensions, one being spatial dimension, which kind of encompasses the visual acuity and contrast sensitivity. The next one is the spectral dimension that encompasses color vision of animals. And the third one is the temporal dimension, which kind of takes all the changes in time in the first 2. And how we see these things depends on how fast our vision is, basically. Changes in time really affect how we see things, and it can aid vision or it can hamper vision. First, I'm going to talk about aiding of vision. So here what we see is a phenomenon called motion parallax. It helps us, when we move in our surroundings, to understand depth, so we know what distances things are at and which kind of sizes they are. We are also very sensitive to any kind of change in the field of view, so if there's motion or flashing on off, it is very conspicuous to any visual animal, basically, or should be at least. Thirdly, motion helps us understand form, so even if something is occluded partly, we can still deduct what is the shape behind the occlusion if we get synchronized motion of this object. In my research, I was interested in how dynamicity affects the detectability of signals, and I did this experiment using my study system, the Urama butterfly, or the common grass yellow as we know it in the common tongue. So a little bit about these animals. They are visual, and they are highly motivated by colors. So they will use colors to find, for example, nectar sources. And we also know that males are extremely motivated at finding mates, and they will go and inspect any kind of yellow thing in hopes of it being a mate. So just a little bit more about their vision. So they are what we call a tetrachromatic. So they will see things from very short UV to deep red. And as I said earlier, they have a pretty bad vision. So in order for this map butterfly to actually see a mate properly, even in a rough shape, it needs to be like 10 centimeters away. If it goes a meter away or 2 meters away, it doesn't really see it very well, whereas as humans we forget this because we have our supervision and we keep seeing things well in different distances. But what they do have is a pretty good motion vision. The animal or the butterfly vision is 2-3 times faster than human vision is. So for them, if they can't see an object by seeing the form of it or having enough color contrast, motion can help them a whole lot in seeing what is going on in there. So in my experiment, I used the mate search behaviors of the males to see how dynamicity in signals affects the detectability of signals. So I made this little model and I watched how many males I would get to approach the model and from which distances they would approach it. And this is how my model looked. So it's just a little dot. It's a paper dot that rotates around its own axis, and even to our superior human eye, it kind of looks like a butterfly, doesn't it? It's fluttering, it's flashing on off. And the males were impressed. They would come and court it a lot. Yes. So I did this stimulus. I presented this stimulus in 4 different treatments. One being a static treatment, it doesn't do anything. One is flashing, as we just saw. Then there is movement treatment, but doesn't flash. And together the movement and flashing, as we have in this video. So 4 different treatments. And I was interested in seeing how the flashing, how movement and flashing together affect how it's detected in the field. And in order to actually follow how many approaches I get, we have different methods of recording it. We used a drone to get an overhead footage of it, and we had GoPros on ground level to properly see when we have an approach to the model. And this is basically what I got in this experiment. So unfortunately, the distances of detection didn't really change between stimulus types. My models kind of indicated that motion might help the butterflies to see it from further away, which makes perfect sense, but I couldn't get a good statistically significant difference there. But importantly, we see that any kind of dynamicity in the stimulus will improve the detectability of the stimulus. And another interesting fact is that the flashing actually helped them a whole lot more compared to movement. So, and this is important because we know that movement is actually a significant signal for insects such as like dragonflies or blowflies that can detect their prey or mates by movement, but flashing is kind of novel. No one's tested it, I guess. It makes perfect sense, but you can't go saying that it's conspicuous to other animals if you don't test it, right? So that was what I got from that experiment. I have done another one, and this considers how changes in time hamper vision. So how changes can, for example, hamper our how we perceive a scene is when something moves too fast for us to actually see the patterning. So if something moves so fast that our eye can't sample it properly, the spatial detail will blur, and some animals actually use this as a anti-predator defense, such as this snake on the left, on the right. Which has like conspicuous striping, but when it decides to escape, it will blur. And sometimes it even helps by blurring to camouflage better into its background for some of its predators. Another way of changes in time hampering how we see things is flash illusions. So when there is a fast-moving object and it's flashing at the same time, there is an actual chance that we misjudge its location in space. So this is an illusion, and it's relatively recent. Sometime in the 80s, somebody noticed that when they wave a cigarette under a strobe light, they see the lighted part kind of floating ahead of its the location where they think it is in their hands, which, by the way, kind of reveals what they've been doing. But so I have a version of this kind of an illusion here. So this is a type of a flash illusion. It's caused by, oh no, does it run? Let's see if maybe the next one is better. Yeah. Can you see the stimuli being tilted to the direction of motion? Yeah. So in this case, so we have a background motion that is confusing our motion processing. What is actually going on in here is that when the stimuli appear, they're exactly aligned with the cross. But our brain has difficulty understanding where the actual location is because there's background motion. And so this is something that could potentially help a prey if it was to be attacked and it needs to escape, right? So I wanted to test this, and I did this kind of an experiment with a Eurasian blue tit, which is a wild animal in Finland, and we actually got a paper out of it just recently. So in this experiment, I took wild Eurasian blue tits, and I taught them to peck at a dot in order to get a food reward, and then I would test them and release them after, of course, back to the wild. So this is the experimental setup that I had, and I tested 20 blue tits with 5 different color treatments, and you're gonna see it right here. So the stimulus, again, is a dot that kind of rotates around its own axis, and it can be either the same color or a different color from the other side of the stimulus. Yeah, so I tested 5 different colors: green, light green, blue, and then the combination colors of green, light green, and green, blue. And I tested them with a lot of different speeds. And this is what I got there. So we indeed found a difference between stimulus types. So when the birds did not hit the stimulus, they had most error distance in the green to blue flashing stimulus type. Also, when they tried to catch the stimuli, the green to blue flashing stimulus type needed to be pecked more in order to actually catch it. And this is interesting because now that we see flashing color patterns in nature, we can, and I'm not saying that these flashing color patterns necessarily are anti-predator defense, but we can think that there is a possibility beyond sexual signals or social signals in animals to have something else there going on, right? So yeah. So just to sum it up, in case it's a take-home message. So when there's dynamicity in visual scenes, it can either help us or it can hamper us seeing things accurately. And in case of movement and flashing, it can make things much more conspicuous and easier to detect. But at the same time, it can also induce us to misjudge a location of a target, and it's because of our motion processing getting screwed by the different stimuli. So yeah, thank you.
So here is just. Hi everyone. So I'm Vanessa. Let's just see if. Okay, cool. So today I'm talking about one chapter of my PhD. I'm just about to finish. This week I will submit it. So I'm talking about the brain of spiders. So I'm comparing either the social spiders, if they have bigger brain when you compare to solitary species. However, as I just mentioned at the beginning of the talk, there is a I'm not saying misconception, but there is a general knowledge that animals who live in a group, like social animals in general, they would have bigger brain than solitary species. So I believe this concept, there is just me in the middle. Cool, so So there is this knowledge about being like, if you talk with someone, animals who live in a group, they have big brain, and those who not live in a group. And although probably this concept came from this hypothesis, the social brain hypothesis, where I state that animals live in a group as a society, they require to have a big brain because to live in a group, they need to deal with each other and to deal with complex social interaction. This hypothesis, however, was established with mammals, with primates. And then since then, we kind of generate this conception to all the animals. And I believe this is an issue too nowadays, because for many animals there is no evidence, so there is no studying about comparing actually the brain or measuring the brain. So after this study has been done in mammals, they were also tested, and they found some support in birds. They also was studies in insects, and they also found some supports. However, many other studies in different taxa did find any support. So I decided, and then so also when you talk about big brains, there is also, then I can say a misconception about big brain correlate with intelligence. So then that's just, I think it's a little bit issue, because then we, as human, we put ourselves very high in the thing. So I'm just trying to say that this conception, based in a few studies from the recent years, has shown that actually that's not true, because new other studies has shown that brain mass, or when you compare just the absolute value of the brain, or even some parts of the brain, they are not very good proxies for intelligence, or even good proxy for brain function or behavior. So that's, I'm just saying that we should be aware about that. And I decided to test this hypothesis in spiders. So here in Australia, we have a few spiders. They are social, and they also have relative species that are not social. So they're a very good model to be studied. So here I study the social spiders, the huntsman spiders. So they have species that are social, species that are solitary, and the same for some crab spiders. So to do that, I compared the social one that I just mentioned to two other solitary species, for the huntsman and also for the crab spiders. We studied them in Australia, from Queensland to Victoria. And then something interesting to say that sociality in spiders is quite a rare phenomenon. So we have like almost 53,000 species, and social spiders for species has been described in less than 0.1%. And something that the majority of the spiders, they live in a group as a society. Actually, they build webs. So they live in these big webs here, and then we have lots of generation living there, keeping the web. However, for the two groups I'm studying here, the huntsman and the crab spider, they do not build web. So they live. For the crab spiders, they live in bark trees, like eucalyptus bark tree. And then they just have the colony there. And then if you open, for example, here in the field work, I did, the sound, sorry. We should have the sound. I'm so sorry. I'm so sorry. Oh, sorry again. Sorry. So it's quite fun because, I mean, for me, they love spiders, but I have talked to some people that are quite afraid of them, so probably be a nightmare. So, yeah, so when you open this bark, and then you start seeing like hundreds of spiders coming, and then I can collect the full colony. So for these spiders, for these species, they are the Dilena cancerogens, I think I forgot to mention, but that's okay. So the system is like we have, normally in this colony we have, sometimes we have just the mom and the offsprings, or sometimes I can find the moms, I can find different males, more females, and lots of cohorts, like juveniles in different states. And then for the crab spiders, as I mentioned, they do not build web. They live like in a leaf nest, like put lots of leaves together, and then they stick the leaves together with web, but it's not like a full web like for capture, just to stick them together. And the colonies, I think I do have a video. Yeah, a small video. Normally the colony is just the mom and the offspring, the babies. So, something that I should mention that different from insects, for insects we have the head, then you have the thorax, then the abdomen. And here in the thorax, we have the legs. So different from insects, in spiders we have the cephalothorax, so that's this bit here, when you have the head and the thorax fused, and then you have the legs coming from this big part here, the cephalothorax, and then you have the abdomen. And then in the abdomen, just for fun, we have the heart. So we can see here, if you be patient, next time you say spider, you can see the heartbeat. It's cute. So the brain and everything, like the central nervous system in spiders, are located here in the cephalothorax. But it's together with muscles, together with venom gland, together with lots of things. So it's a little bit hard to study the brain of the spiders because, as I said, everything together here. But just to go to the most interesting part, that is the results. So how I measure the brain. So I did some, we call like micro CT scan. So it's very similar with us when you go to the doctor and you do MRI. So the doctor get this kind of image from your body. So the same thing I did for spiders. So once I have this image, I can then delineate the areas of the brain or the areas I'm interested in, and then I can get a 3D mode, a 3D reconstruction of the things that are inside. So here, this big green thing is the venom gland, and then we have here the central nervous system, and then a part of the ventral nerve cord, then we have the proper brain, and inside of the brain, we have also the visual neuropils. So as you know, spiders, some of them has big eyes. And I don't want you be very careful, pay attention on the names, but just to mention, inside of the brain, like we do have two important structures in the brain, like the mushroom body and the arcuate body. They are very important, like high center of integration of the information, and they are related to the learning and the cognition of the spider. So they are important structure to measure and to observe and for comparison. So we found that when we compared the overall size volume of the brain, like the overall size of the central nervous system of the spiders, in huntsman spider we did not find difference. However, when you look inside of the brain, so here in the graph, you see like the proportion of the brain, and here we see the structures, the individual structures. So when you compare the proportion of the structures inside of the brain, then we found that these two structures that I just mentioned before, they are, so just comparing the structures inside the brain, these two structures, they are actually larger in social spiders when you compare to the solitary ones. In crab spiders, when you did the same analysis, this structure does not differ actually. There is no difference between these two structures. And the only one that's bigger inside of the brain actually are the visual neuropils. So we're looking at two different spiders, two different systems of sociality, and then you can see now difference inside of the brain. So we're trying to understand what's going on. So to try to understand what's going on and why you can see this different pattern in these two groups of spiders, we observed that, as I mentioned before, in social huntsman, we do have a society that is more complex. We have not just the mom and babies, but we have like older adults, lots of males, lots of females, things going on there. And then here in crab spiders, just have the mom and the babies. But something that I didn't mention before is in, oh sorry, do you mind just put my face there? So in huntsman, we do have what you call a kin recognition. So the spiders, individual inside from the colony, they can recognize members from the same group, being very aggressive toward co-specifics from outside of the group. So they do have a kind of system of recognize the members by using chemical cues. And so this is quite unique in this group of spiders. And for crab spiders, we should know, so more studies need to be done. when you come to trying to understand what's going on here relating to the visual system, for crab spiders, not just the social, but all the solitary species too, they all diurnal. So they sit in the flower waiting for bees or for some pollinators. And then we're trying to understand if they all come from the same diurnal system, why they have different investment in visual neural pills. And then when you look at the literature, we found that some animals who predate at night, for example, when they live in low light environment, they normally have larger eye diameter. And then we look at our spiders, and they both diurnal, but this one, the social one, they live inside of this leaf nest. So the environment inside of the nest is a little bit low in light. So then we're correlating, we still need to do more studies, but we're predicting that to be inside of this low light environment, maybe they have larger visual neural pills. That will explain a little bit our results. So just to wrap up, so spiders are excellent models, I think everybody's agreed. And then we did see variation in the brain of the phylum, as I show you here. There is a variation in the brain. Some of this variation allied with for the social brain hypothesis, but because we didn't see the difference in the two taxonomic group, we don't think the social brain hypothesis is actually, we think the social brain hypothesis is very simplistic, to be honest, to explain this variation. And we believe there are like more other factors in the biology and the society and the light, everything that is related could be another factor, and being sociality is just one of them. And then we believe that more studies, like for example, counting neurons, will be a very good additional, so then we can proxy more with correlation and integration system. So I think that's it. Thank you very much.
Hello, everyone. My name is Marilia. Some of you might remember me from the Skeptics from the pub that we did in the beginning of the year. And I'm hopefully, and that time, I did a little bit, which is called, is this a butterfly or a moth? Because I was trying to convince people that moths are not just ugly butterflies. As you can see here on the right, we have a beautiful butterfly. And on the left, we also have a beautiful moth. So I'm here to show you that this is not my only party trick. And hopefully you'll play along with this. So this one is a game that I like to play with some of my friends, which is called, Is This a Bee? And like our young friends from yesterday, I decided to pick an easy example to start at first, where you can raise your hands up if you think it is a bee or not. Here we go. So is this a bee? Yes. Absolutely right. And you can tell that the giveaway is because bees usually have opposable thumbs, so that's very easy one, right? Now let's try something a little bit more difficult. Is this a bee? Yes. Yes, most of you think it's a blue bum bee. It's one of my favorite ones here from Australia. How about this one? Yes, no, this is not a bee, it's actually a fly. And you can tell by many different things that you can see in the future, but for example, the antennae are different than bees' antennae. This is one of the giveaways you might want to pay attention. How about this one? Bee? Fly, very good. So you saw the antenna and you're like, there's something wrong, but there you can also see that, for example, this one only has 2 pairs of wings, which is not usually with bees. How about this one? Bee. Yeah, you can. The colony is a little bit of a giveaway. So this is a queen bee. It's a little bit, the abdomen is a little bit larger than normal bees. Let's see this one. Who thinks it's a bee? Some people? Okay, it's also a fly. It's a little bit of a better fly because the antennae are very similar, but you can still sort of tell with the general body shape. Let's try this one. Bee or not? Bee. So this is an orchid bee. It's very common in South America, which is where I'm from. And they're usually with these beautiful metallic colors. This one. Bee, fly. This is a bee. It's a stingless bee. And from another group in South America, because you can see I'm a little bit biased. What about this one? Fly. Okay, very obviously a fly. You guys are getting better. Very good. How about this one? Also a fly. This one? Bee or fly? It's a beetle. So, tricked all of you. And for those of you who are a little bit more specialistic entomologists, you can see that there's this pair of wing here, which we call an elytra, which is characteristic from beetles and nothing else in the insect kingdom. This one. Also fly. This one? This one is a bee. So this is a carpenter bee. It's this one? Bee as well. Very good. It's a fly. This one. Beetle C. Some of you are picking up. Very good. This is a hoverfly, again, to show that moths are not just ugly butterflies. I have to keep on theme. Fly. Also metallic color, but you can see it's clearly a fly. And lastly, we have this one. Wasp, exactly, but it's very similar to a bee, isn't it? How can you really tell? As you ponder on this question, let's go to notes. So you can see that there's a lot of things that look like bees and are not bees. So what really is a bee? A lot of people like to say that bees are just vegetarian wasps. And I think I heard one of you say that too. And this is actually not true, because there are some bees that eat meat. They're called vulture bees. You can, again, guess why. And we also have wasps that, a lot of them are omnivores, but they often feed on nectar and pollen when they're adults. So bees are not necessarily vegetarian wasps because the diet is switched. So what else do people like to say about bees? Bees are social wasps. But again, we have a group that is called social wasps, so it's a little bit dodgy. And we also know that most bees are actually solitary. So this doesn't really hold up as well. Bees make honey. This is a good one that the internet will tell you. And we also have some social wasps that make honey. So you won't find this commercially, but they use it to feed their babies just like bees. And as well as the solitary bees don't make honey. So we only have one group that makes honey, which is usually the social ones. Is it because bees have queens? This is another one that I heard. And we do have queens in wasps as well. And the difference is that in a queen wasp, it usually wins its throne by dominance. So the most aggressive wasp will rise up to the colony and become queen. As though in bees, it's more like humans, where you're born into royalty, you know? So, and again, solitary bees wouldn't really have queens, or would they all be queens? And I'll let you decide with that one. So this is my favorite one, that bees are nice wasps. And everybody loves bees, right? They pollinate, they're cute, and everybody seems to hate wasps. They want to get rid of them as quickly as possible. Yet, when we look at the data of people who get hurt by bees and hurt by wasps, we see that a lot more people get stung by bees than wasps. So I think they're not really that nice. And one of the reasons that is because wasp stingers and bee stingers are actually quite different. This is one of the ways. So bee stingers, they sometimes have a fold in them, sometimes they're serrated, and they're like this because they stuck to you. You often hear the idea that the bee will sacrifice itself to defend the colony, and when the bee sting sticks to you, it sort of removes a little bit of the abdomen with it, and then they can just dehydrate to death, which is very interesting. Wasps don't have this problem. Their sting is very straight, which means that it can sting you multiple times. Yay! So this is one of the difference. And again, when we look at, even though wasps can sting you multiple times, when you look at the data, we see that most of the damage to humans is done by bees, not wasps. And in fact, I heard somebody say, this is may not be true because I have no reference, but that bees are the most dangerous thing to biologists on the field. So watch out, my friends. So are bees just wasps? In a way they are. So they comprise this group called Hymenoptera, and where we have lots of different kinds of wasps, and then here in the end we have bees and we have ants. So they're like cousins of wasps. And so there's this group called Hymenoptera, which hymen means basically membrane, and then ptera comes from wings, so they have membranous wings, and a lot of other insects have that, so that's not very remarkable. But on this group, they also have a thing which is called a PTO, where the first segment of the abdomen is constricted. So the body shape of one of these insects is a little bit like a number 8. This is how I learned in school. And some wasps are particularly very good at this. Look at how elongated this is. This is just the first segment of the abdomen where the nervous system and the digestive system have to pass through, which is fascinating. So if you see something that has this restriction, it will either be a bee, a wasp, or an ant. Speaking of, so how do we tell bees apart from wasps? Well, bees usually have tongues. They use this to lick up nectar, and as well as mandibles. And wasps generally only have mandibles. They're also a little bit rounder, so even though they do have the constricted abdomen, sometimes it's more hidden because they're just so round and fuzzy. And they have an adaptation in the third pair of legs, which makes them a little bit flat, and they can use this to store pollen. And in this case, which is the metallic bee, this is an orchid bee, and they collect perfumes from different plants to impress their mates. So we can learn a lot from them, you know? And, but then what about ants? They're in there too. Things also tend to look like ants. So take a moment in this picture and tell me how many ants do you think there are? Well, there are 4. These are the real ants. Most of the other ones are either spiders, or we have here a mantis, an assassin bug, this is a cricket, and we have a few beetles. And it's also fascinating because we have this group of animals, which are bees, wasps, and ants, that a lot of things want to look like them. And why do a lot of things want to look like them? Well, you might have guessed, it's because they're assholes. And so what we have is that they're very dangerous. Nothing really wants to pick up a fight with an animal that can sting you and sometimes live in a colony, because then multiple animals can sting you, you know? It's very nasty. And wasps are top predators of the animal kingdom. Well, they even hunt spiders, which people are afraid of. So yeah, like wasps, they're nice too. And so what we learned from this part of the talk is that there is a very big advantage into disguising as a bully, as social media has tried to teach us for years, right? But is this the only behavior that is worth copying? So as you might have guessed by my shirt, I don't actually work on bees or wasps. I work on butterflies, which are the exact opposite. They're very nice. They have no stings. They have no claws. You know, they are what I like to call the nature equivalent of a flying hamburger. So let's try to see something eat them then. This is an experiment that was done many decades ago with a blue jay, and it finds this beautiful monarch, it tastes it, and then it thinks for a moment and throws up. Not the nicest hamburger out there, right? So what happens is that a lot of butterflies, like monarchs, they have toxins in them. Monarchs, for example, have something called cardenolides, which will stop your sodium potassium pumps from working properly, which basically means that they can give you a heart attack. So, you know, not the nicest thing to eat. And because they can do this, a lot of other things like to copy them as well. So you don't have to be a bully. You can, you know, have your quiet defense in order to do this, which is why I think butterflies are quite nice. And what I work is what kind of patterns are interesting in eliciting this response. And this is because it works like this. First, you have a beautiful bird who thinks that life is nice, and then it sees a stimulus. This is based on my own experience. I really don't like coffee. And it approaches the coffee. And then it's like, this is nasty. I don't want to have it again. So next time it sees the coffee, it will reject the coffee. And this is a process we call aversive learning. And I work in what kind of patterns makes aversive learning work. And in order to do this, I created this game that I hope that you all play with me right now. So in case you're worried, we don't collect any personal information. And you can scan this QR code and play a game where you have to find the butterfly on the screen. And I'll just give you a little bit of an example while you get your phone out. But actually, I need to leave the QR code there for a minute while you scan. And if you're seeing this online, you can also type this on the computer instead. Sorry. I see people have started playing already, so good on you. Just an example, there will be, for those of you who are not playing, there is a background and there's a butterfly hidden, and you basically need to find the butterfly. So I will give you a few minutes to play. I think 3 minutes should be enough. And then I will compute your results while my friend talks to you also about butterflies. So play a little bit, and I guess we'll be right back after 3 minutes.
Yeah, I'm waiting.
Don't wait, son.
Okay. Now? Okay. So hi everyone, my name is Mathilde. I'm a first year PhD student and I really love butterflies. What I really like to is going out. So I've been explaining to a lot of new people what I've been doing for work, and I study butterfly behavior. And so when I say that, what I hear a lot is, but how do you study them? They only live one day, don't they? And that's true that butterfly don't live that long actually. On average, the adults live 2 to 4 weeks. So it's not that long, but it's still much more than one day. And there is some butterfly that can live pretty long as well, like the brimstone butterfly or the mourning cloak that can live up to a year. So to live that long, they need to be able to learn, to think, to make calculated decision, and to remember where they can find their resources, their food plant, and where they can lay eggs. So butterfly and insect in general are actually much smarter than what we used to think. That's been studied a lot in the past decades or so, especially on honeybees, and it's been shown that honeybees can actually learn how to count up to 4 only, but that's still pretty impressive. And they can understand concepts like above and below, or same and different. And bumblebees are also pretty smart. In the lab, they can be taught how to pull a string to get a nectar reward, and they can also show other bumblebees from the colonies how to do it. So that's called social learning. And what about butterfly? Butterflies are also very smart. They can learn to associate specific color with food, with a reward. They can find their way back home when they're put in an unfamiliar place, which is something I struggle to do. And they can also copy the behavior of other butterfly when they don't know what to do, like something we would do, like look at our friends when we don't know what to do. So that's about what I studied a couple of years ago when I was in Belgium. I studied the impact of habitat degradation on the behavior of the wood-white butterfly, Leptidea sinapis. And what we wanted to do that because habitat degradation is something that happens very fast. It happened too fast for evolution to actually act and make them adapted to their environment, because evolution takes usually a lot of generation to produce noticeable changes. So if they can't adapt really through evolution, maybe they can adapt their behavior and become better learner and learn new ways to use their environment. So what is it important to know that? Because it helps us to know which population of butterfly might be more at risk of becoming extinct. Because being able to learn and to adapt your behavior to new environments is something that that can increase your chances of survival, but you need specific brain structure to be able to do that, and those brain structure consume a lot of energy. So this energy can't be used for other things like reproducing, for example. So if we have smarter butterfly that reproduce less, if something happened to our population, let's say winter frost, and a lot of butterfly dies, then if they can't reproduce enough, then this population will take a long time to recover, or maybe it won't recover at all. So what we did is that we went around Belgium and we collected butterflies, some of them from very degraded habitats that have very small and isolated good habitat patches like that, and some butterfly from well-preserved habitats. And then what we did, we released them in an arena with plants where they could lay their eggs, and also flowers so they can feed. And in some cases, we put other fake butterflies with them, and in some cases, they were on their own. And then we spent some time recording their behavior, where they go, and all that. And so we found some very interesting thing. So what we found the most interesting was that when there was in blue is when there is other butterfly in the cages with them. So when the habitat become more degraded, they tend to stick together. So they explore the environment less when there is other butterfly with them. And also they spend more time resting at the same place. And we saw the opposite when there is no other butterfly, when they are on their own. So basically that means that butterfly from degraded habitats stick together, and that butterfly can use other butterfly to gather information about their environment. And the way they use this information is dependent on the type of habitat they're from. But why would they do that? So when they are in the degraded habitat, the resources tend to be very rare, and they're very grouped together. So staying where other butterflies are make them sure that they stay where the few good resources are. Well, when they are in the good habitats, the good resources are a bit everywhere. So staying together means more competition. So when they see other butterfly, they would rather go on their own way to find their own resources and avoid to compete with the other butterflies. So why is it important for us to know that? Is that this kind of behavioral response could increase the extinction risk. If butterflies stick together, then they have more chance to breed with relatives, and so that's inbreeding, and it's quite bad for the population. It's reduced the genetic diversity of a population, so if something happened, there is less chance they will recover, and it also increase the risk that they express bad DNA mutation that are bad for their survival. And they also have less chance to discover new resources, and so the total amount of resources available to the population is much smaller than it could be. And so that means smaller population, and smaller population means more risk to be extinct. And so that's also especially true because one of the things we also found was that as the habitat gets more degraded, butterfly lay less eggs, so they're potentially reproducing less. So the take-home message maybe for this presentation is that butterfly have very flexible behavior, and they can adapt to their environments by changing their behavior, and they can also use information from other butterfly to display the most adapted behavior. But this change in behavior can increase their risk of going extinct as well. And overall, what I think is important to remember is that insects in general are much smarter than what we used to think. Thank you.
Hello again. Just a few more minutes of my voice, I swear. So, welcome back. I'm going to show some of the results of the game that you played. So first, this is how well you're doing compared to the average of people. So we have over 1,500 people play this game already. It's part of my PhD research. And in red here, we have how many butterflies the previous people found proportionally. And in blue, it's you. So you can do a little bit better. So don't worry, try again. It will only help me. And this, which is it's the most interesting one, is what are the butterflies that are harder to find for some people compared to the other people. So in the bottom here, we have the overall results for over 1,500 people. And we can see that the blue ones seem to be more conspicuous compared to easier to find in this case compared to the brownish ones. In the conference we see a less clear pattern, which is why we need a lot of people to play to gather data, where you have again sort of like grayish butterflies all over the place. Now, why is this interesting? What am I trying to do with this? Well, as I said before, I study how toxic butterflies are. So for this, I got a lot of butterflies across Australia, and I did some basic toxicity assays where you have here some preliminary data of my PhD. In the top left is the most toxic butterfly we found, and it just keeps going down. And we measure this by extracting toxins from the butterflies, and we test them on water fleas, and we basically measure how many of them die. And you can see that the colors are sort of all over the place. And when we compare this to the preliminary data from the game, to which butterfly is easier to find than the other, this is how it looks like. So it completely re-scrambles it, and where we have a clear trend where the more bright butterflies, so the ones that have white on them, for example, seems to be more conspicuous than the darker ones. So basically, what can we take from this is that we really don't know what kind of patterns elicit aversive learning yet, which is the process to which something copies another organism that is dangerous. And we're looking into more different things, like how does the butterfly pattern and not just how easy it is to find takes a look into this, but maybe that data will be for the next time. So thank you all, and we have a few minutes for questions, which you can do for all of us.
Yeah, thanks. So that was fantastic. Just wondering whether maybe some predators are seeing in a different range to what we're seeing, and therefore they're picking up on something that we're just not seeing when we try to distinguish.
Hi, thank you for the question. Yeah, so birds, like Sunny mentioned, they're also tetrachromats, so they see the whole color range, and this is what we assume that preys on butterflies. But I have taken pictures in the UV of all these butterflies, so I can know how well birds see them. And what we usually find is, for example, white. What we see white usually has UV, and what we see blue usually has UV. But the pattern itself doesn't change that much when we compare the UV photos of them with the visible photos. So it is a proxy, but it's a very, very good proxy.
Any other questions?
I have a question.
Up the back. Okay.
Is the fundamental purpose of your research to protect this particular species, or are there things that you are learning that you're able to translate into the nature of brain function, the nature of sight, the nature of movement? You know, I'm interested in the directions that your research can go in.
Brain research, I must say.
No, he said in general.
In general.
In general.
Yeah. Brain research, so.
Yes, so about conservation, that was you're asking something? Yeah, that's if I'm sorry. Yes, when you, for example, when I was surveying these spiders in nature, I noticed that because of us, of course, we are destroying the environment. So most of the areas that are used to find these spiders, for example, in public parks or even in national parks where you have the eucalypts, and mainly in the general parks, we are just destroying all the trees because these spiders, they're very related to the bark trees. And then if you go next time to the park, you can see people just sometimes walk in the park and remove the bark trees. So we're just removing all the retreats for the spiders. And they are very related to the environment. And then in most of the areas you go, we don't have this environment anymore. So I have seen from my studies when I started in 2021 is nowadays I can find less and less. So yes, I do have bringing this brain concern, like to understand how the spiders, how it works, because we didn't know in the past how the brain was working, like how the nervous system of spiders is or something. We're trying to, of course, bring aware of the importance of these spiders, and also there's a few studies in a few years showing that spiders maybe can dream. So maybe, yeah, so they can sleep. And then when they sleep, they do some kinds of, when we are sleeping, or for example, our dog or a cat, they do some eye movement, like when we sleep, we do like this ERN movements in the eyes. And then we also, the dogs, for example, they curl a little bit when they're sleeping. So 2 years ago, a few of the researchers in Europe, they showed that some spiders can do that too. So now we are interested to understand more and more about the spiders and go to more the intellectual thing. And then maybe in some years, we can find out they are sentient animals, and to be more protective. So for example, I think we were discussing a few days ago, yesterday with someone in the dinner, about there is no ethics for spiders, for example. We don't need apply for ethics to study them, different from vertebrates or, for example, octopus, that we need to put ask for permission to study them. But maybe in the future, understanding more about some conscience about the body or something, that we can change the law. So I hope I have replied.
I'll just give a really short answer to that too. I think many of our research actually spans further than our study species are. Like in my case and also Mathilde's case, it's clearly we map something with our study systems, but for example, the phenomena that I'm looking into, the vision stuff that I talked about, it spans beyond, like the things that I talked about applies to compound eyes and the lens size of the human. So we are talking about wider concepts. We just used study systems that we can actually tap into that.
Probably all we have time for. One more up here. I'll be super quick.
Hi, do you have any comment about the difference in social brain structures of arachnids and arthropods to mammals, in that herd animals actually have smaller brains to pack or solitary predators?
Yes, so when you compare something that new studies in the brain from spiders has shown, actually the brains of the spiders, they have small areas inside of the brain that before we didn't know. Like in the past, we thought like the central nervous system of the spiders were more like just one ganglion or something. But nowadays, thanks to the technology, we can see inside and see they're like packed areas inside the brain like us. So we have the neocortex, we have all the other areas, and then now we find out some areas, for example, for humans, you have the neocortex where you have more of the neurons condensed inside, so then we can understand a little bit why we are humans have disabilities that other animals do not have. So the same for the spiders. Now we are just starting to understand how the nervous system or the brain works. So then maybe we can, in a near future, correlate more of the behavior with the information we have from the brain. So I hope I have it.